Away with this one. So, of all the projects, so you know, I mentioned that I said, what do you guys want to do? So, Kiteara says, I want to make bacteria smell like a rose. And I think part of it may have been because she was back in the corner with the stinky bear. I'm not sure. And so she had a certain motivation from that point. But the concept, at first I was like, that's completely insane. You know. And then we started looking at it, and it turns out there's this huge area of research for people trying to change the smell of things by altering the gene expression. And so, you know, it turned out to be something really, really cool in terms of the concept. Very, very challenging in terms of the actual implementation. And so I'm going to let her talk about it. It's a neat project.
Okay, so my project, this is the title from my paper, so I'll explain this. Creating a transgenic organism by introducing Rosa Rugosa RNA into Escherichia Coli to produce a scent. Basically, we took a gene from a rose, a Japanese rose, or commonly called here a beach rose, and put it into E. coli to try and get it to smell better. So, the basics of E. coli. It's a very simple organism. It has prokaryotic cells, and all of its genes are contained on a singular circular chromosome. So it's very easy to cut it and put something into it. If you want, that's why it's so popular and a lot of microbiologists like to play with it. Its entire genome has been sequenced, so they can look at it and say, all right, this is where we want to put this gene, this is where we want to put this gene. It's very simple, so it easily accepts new genes. It doesn't have a lot of things that will interfere with, like it won't reject a new gene if you put it in. Other strains are harmless, except to your nose. It smells like rancid creamed corn. It stinks. And every single person in the class knows it stinks because I made all of them do the sniff test. A problem with the plant DNA, and the reasons that most microbiologists avoid it like the plague, is that it's eukaryotic cells, it has multiple chromosomes, so every kind of gene is contained on 2 strands of DNA, and you have to have both of those to be able to do anything with it. The cell walls are very hard to break open. The DNA is spliced, so it's set like, there's 13 genes in a rose that produce the smell. They're separated. So you have to take those different ones out and put them together to be able to put them into people. The cells contain metabolites that contaminate the DNA that you actually get out of it. And there's not a lot of history of working with plant DNA, and this project actually took Dr. A and Michael to go do the research and figure out how to do it, because I couldn't find anything I could understand. So the idea behind the project is to isolate one gene, or the strain of genes, of the mature mRNA that has to be purified and amplified to produce cDNA, which basically you take the RNA and you clone it with the DNA so that you have the full strand. cDNA then has to be put into a shuttle vector and ligated into the bacteria. Basically, you cut open the bacteria, you put it in. That's all that fancy stuff. Only some of these bacteria will accept the gene, and even if they accept it, they might not have any difference. So the method, I'm just going to explain this in layman's terms because this is a lot of gibberish. So Dr. A actually brought me a sample of his rose at home, which I had to identify, so that took a while. But it's a Japanese rose. We took 10 samples of that. We treated them with Triazole, which basically just was an RNA extracting agent. They were mixed in a fast program. Yeah, I lice the frozen tissue. We beat the crap out of them so that the DNA would come out of the cells. That's all that is. They were treated with chloroform to separate the RNA, and then we took that out because we don't want the protein and we don't need the DNA. So it was cleaned, and when I say we put the ethanol in, we had to put it in, centrifuge it, so that all of the actual RNA was like pelleted at the bottom of the tube. You had to take the ethanol out and then put in nuclease-free water, which is this little tiny vial of really expensive magic water. Yeah, don't drop it. We put it in this thing called a spectrophotometer, which basically measures the concentrations of basically stuff in the solution, and then something called the 260, 280, which measures like the purity of the RNA sample. And 4 samples were chosen based on their purity and the part of the plant they came from. We took one that was from the leaf, one from a stem, one from the petals, and one from an actual rosebud. And most of the concentrations were pretty high, and the purity, to give you an idea, 1.9 is like absolute perfect purity, and my samples went from like 1.4 to 1.8, so they were pretty close. These samples were treated with this magic primer that Dr. A had to make, or
purchased.
yeah,
Had
had to find,
yeah.
called OOMT2, and then run it through the agarose gel, so the gel electrophoresis, to isolate the correct gene. To give you an idea, I think yep. So this magic little thing is the actual sequence of genes that we needed. They had to be the correct size, so that's why you do the gel, is it separates out all the genes by size. The gene was inserted into the shuttle vector PKS promo, which was then digested with the enzyme small ones to create the cDNA. Yeah, that we took that out of the gel and processed it with a lot of stuff and ligated it into the E. coli. And it was put in an incubator shaker, and that's a very self-explanatory title, an incubator that shakes it. The sample was then transferred onto 7 auger plates, I think it was Luria auger, and allowed to grow colonies overnight. And there's actually pictures of these at the end, but the colonies that took the gene grew and were white. The ones that didn't grew and were blue. So we could actually look at those and say, okay, we took 2 white, 2 blue, and 2 that were kind of like blue-white. And they were like, and those were streaked onto 6 new auger plates, and those were growing. So, and then I made everybody do the blind snip test, including the teachers. Yeah. So, the results, this wasn't really one of those projects where it has really clear-cut results. It has, like, the results are, the rose gene was successfully extracted and purified for use. So we actually got the RNA out. That was the first mountain to climb. The gene was then transformed successfully into the E. coli and expressed in over half the colonies, which was actually a really good success rate for trying to put eukaryotic RNA into a prokaryotic cell. There was no discernible floral scent, but there are possible explanations for that. 3 main reasons. There are 13 genes that are required to make a rose's scent. So there's a possibility that we might have missed some genes, or that they didn't all carry over through the whole process. Another explanation would be that we needed a different media for the bacteria, because they need basically to have different ingredients to create the different things to make the smell. All a chemical process that I don't understand most of. Yes. And then the third possibility is that maybe E. coli just doesn't have the genetic ability to produce the smell. Conclusion, project was successful. Just because it didn't make the smell doesn't mean it was bad. Shows that there is a real possibility to produce a transgenic organism that carries a new smell. And it proves, not supposed to use that word, but it proves that it is possible to extract plant DNA of good quality with standard lab equipment. And it also shows that given the correct conditions, eukaryotic genes can be transformed into bacteria. And this is actually a picture of my 7 plates where we first grew them. You can see the ones with the circles are the 6 that we chose to actually grow for the snip test. And then this is actually a good picture where you can, let me get out of the way. You can actually see, this is a dark blue one, this is a light one, here's a in the middle one, so you can see the color difference. So, yeah.
You guys have questions?
You said that the E. coli that accepted the genes turned white. Why would they turn white?
The,
Is there something else you transferred into them?
yeah, there was some of the stuff that we put into the mix at some point. Basically, when E. coli processes its sugar, like the gene that causes it to process sugar, part of it will cut open the sugar molecule, and it releases a blue dye. When we put the new gene in, it actually interferes with the process, so it won't release the blue dye. So it's not that the E. coli turned white, it's that it didn't turn blue.
Gotcha. Thank you.
Anything else?
Yes.
If you needed a new name for it, you could title the talk "A Rose Like Any Other Name."
On the smell test afterwards, was the E. coli less pungent?
Actually, I had a lot of different results on that. There was, like, a lot of people said it's not as bad, and then a lot of people said it still smells just the same. But then it was a classroom full of teenagers, and most of them didn't want to do it, so it was horrible.
I was one of them, so I like to go big with the ones that did take the G and A were rancid, burn your nose here. Then the blue ones that didn't accept the G, they weren't as bad.
So she actually made me know what you're saying.
It smells worse. It's very different, but.
Yeah, well, I mean, that's kind of one of the catch-22s, I guess. You throw the genes in there, and whatever they can grab a hold of and make.
You threw the scent gene in, so it played with the smell.
Yeah.
So we don't have to dump stock into the speed stick or anything just yet.
Summer Ventures 2014 here at ECU. This has been an awesome time. I've been totally blown away by what I've seen, not just in the classroom. You know, we all know that you're all academically exceptional and gifted, but last night I went to the talent show, and I was blown away by your artistic talent. You know, I play piano, but you guys can play the piano. I was blown away. The voices, the poetry, it really blew me away. So that was one highlight I didn't want to mention. There's another highlight. You can go to the ECU homepage. Let's give it up for archaeology, right? There's archaeology. They have been highlighted on the ECU News. There's a nice, there it is, right there. There we go. Give it up for archaeology. Feel free to go to our website, forward it to all your friends and family, donors. That's always a good one to send them to. I do want to give a few acknowledgments regarding the program. First, I want to thank the faculty. The faculty is the lifeline of this program. So I do want to acknowledge, starting, I'll just go by class archaeology, Dr. Charles Ewing. Is he here? Hey. And he has two assistants, Ms. Catherine Thomas, is she here?
It's a California wedding.
Oh, lucky her. And Coy Idle. From physics. Physics had all the cool toys. I don't know if you saw that, like the bionic hands and everything. That's really cool. Oh, I'm sorry, it was engineering. Yes. I'm sorry. Dr. John Kinney, who is, I just learned is in Wales. Yeah, he's in Wales. And Dr. Mark Sprague. And his assistant, Mr. Wilson Hawkins. DNA fingerprinting, which I learned you can actually make bacteria smell better, which was Dr. Eric Anderson. And his two assistants, Mr. Michael Rubens and Mr. Joe Blackburn. One course wasn't part of the research scope, but the first 2 weeks of didactic, we did have data-driven decision-making by Dr. Jason Brinkley. He is not here. He just sent me an email saying that something came up today. But I do want to acknowledge him and give him a round of applause. biomechanics, I'm sorry. Explorations in engineering, Dr. Ricky Castles. And Dr. Zin Zhu. We put away our school book. And their assistant, Mr. Brandon Watson. He's also put away. There are also a wide host of mentors from the medical school. Are there any medical school mentors here today? No. Well, I do want to acknowledge your service, even though you're not present at the moment. Now for staff. This is very, very important. Staff, what I mean are the RAs and our admin. They're with your students all throughout the day, and they're really their caretakers during this time. So I want to acknowledge them first. Mr. Michael Corbett. Thank you for your service. I have some goodies for you. Ms. Linda Brown. Ms. Tiffany Burgess. Jacques DeBose. I'm going to mess up your last name. I'm sorry. Alec Eisenberger. And he is Batman. Paul Zander. This year we did a little something different. We hired a junior RA who was a previous Summer Venture student, Ms. Ayushi Patel. Unfortunately, she did have to leave, but I did want to acknowledge Ayushi Patel, who was a great help to the RAs this year. Now for our administrative staff. There's a lot of work that goes on behind the scenes that you don't have an opportunity to witness. We do have an awesome student this year who has probably contacted you, who's been organizing all the information as it comes in, Miss Alicia Gerkins. When I went down with my back surgery, one professor stepped up and really helped us out with Summer Ventures, and she has just been an awesome addition to the program, Dr. Maureen Grady. I'm not sure if you recall, during the opening, I said one particular person is the heartbeat of the program. Fortunately, she could not be here. But she is back now. This is the first time you can put her face with the name of all the wonderful emails, Ms. Cheryl Miller. And if I left out anyone, I'm so sorry. It is a condition of my mind and not my heart. I truly apologize if I left anyone out. Now, for the moment you've all been waiting for, certificates. So I'll bring up Michael Corbett, and he will present the certificates to us.
First and foremost, let me apologize if I do anything with your child's name that is inappropriate as far as like if I totally disagree with your child's name. I apologize. I have been working on them though, and I think I have them. So, but I am working on lack of sleep, so we'll see how well I do. Okay, we're going to start with Windows Group, so if we can get Windows Group to come over here, please. Aaron Ansbro. Allison Chang. Alina Helm. Madison Ketchy. Diamond King. Rashika Lugendron. Janelle Neal. Kiteara Resnick. Amanda Smith. Regina Toby. And Jasmine Wayne. And that is Tiffany's ruling. If I could get my representative over here, please.